Hello everyone, welcome back to embryology sessions. In previous session we have learnt about the gametogenesis in males and females, that is spermatogenesis and oogenesis, and we have seen even the cell cycle and menstrual cycle as well. So here today I am here to talk about first week of development. So first week of development, that is the beginning of the development of embryo. So where we see the formation of zygote. So in first week, it extends between first day to sixth day. So the fertilization happens on day 1, and then cleavage where the fertilized zygote starts dividing, which happens during second and third day, and compaction where a group of cells will compactly form surrounded by a zona pellucida layer, which is called as compaction, and this event happens on day 3, and the formation of blastocyst at the day on day 4 and initiation of implantation on day 6. So these are the major events happening during first week of development. Starting with the fertilization. So fertilization, it occurs where we know at the end of spermatogenesis sperm is produced, and at the end of oogenesis secondary oocyte is produced. So the rupture of ovary we can see here, which is called as ovulation. So we can see the stages of ovulation. So in females, an acute raise of LH, that is luteinizing hormone, triggers the ovulation, and it forms the development of corpus luteum, means the remnant of follicle will form the corpus luteum. So LH surge occurs usually on 34 to 36 hours before ovulation. So increase of luteinizing hormone occurs between 34 to 36 hours before ovulation, and some authors mention a wide range like around 24 to 48 hours LH increase would be there before ovulation. So luteinizing hormone is produced from gonadotropic cells of anterior pituitary, so anterior part of pituitary gland. So after ovulation had occurred, the oocyte, that ovum, it remains fertilizable for 48 hours, and although the chance are almost around 18 to 24 hours. If no fertilization occurs, the oocyte will degenerate, and that is before 12 to 24 hours after ovulation it will start, it may undergo degeneration, and with which the ovarian cycle, a number of primordial follicles or primary follicles begin to grow. So that was about the ovulation. So every ovarian follicle, we see the number of primary follicles, they begin to grow. So we can see the number of primary follicles, they begin to grow to form a Graafian or a matured follicle, and at ovulation the oocyte is in metaphase of second meiotic division, and it is surrounded by a glycoprotein layer called as zona pellucida and corona radiata, some granulosa cells or follicular cells called as corona radiata. And this secondary oocyte along with zona pellucida and corona radiata, they sweep. So the sweeping action of tubal fimbriae carries the ovary. So you can see the fimbriated end of the uterine tube, so which takes up this uh, secondary oocyte. So second meiotic spindles you can see, and we can see the zona pellucida along with cumulus ovaricus, which forms the corona radiata, sweeping along the tubal end, that is the fimbriated end of the oviduct, and it carries the oocyte into the uterine tube. So now that was about the ovulation. So the first event happening is the fertilization. So some active sperms enter the cervical canal within a few minutes and invade the mucous alkaline cervical plug, and which at the time of ovulation it is softened to allow the easier penetration. So once they are through the cervix, so here we can see, so vagina, once they are through the cervix, the active spermatozoa ascend quickly within 1 to 1.5 hours to the uterine tubes, and they may meet in the mature ovum. So all the sperms remaining in the vagina for 2 hours or no longer, they are all killed by the vaginal acidic discharge. And although the sperms remain alive and motile in the cervical canal and the uterus for 5 to 7 days, and in the fallopian tube for 85 hours, and they are usually do not retain their power of fertilization after 24 to 48 hours. So after the 24 to 48 hours of their entry, so till that, fertilization which is normally occurs in the oviduct. So the site of fertilization is the fallopian duct, to be specific ampulla of fallopian duct or oviduct. So the oviduct is divided into intramural part, isthmus, then ampulla, and the funnel like fimbriated end. So at the site of ampulla, that is the dilated part of the oviduct, that is the normal site of fertilization. And suppose anywhere else it gets fertilized, it is called as ectopic fertilization. And because of this narrow opportunity window, the spermatozoa must be present in the fallopian tube at the time of oocyte arrival. So the fertilization occurs in the ampulla of the fallopian tube, and zona pellucida, which is a glycoprotein layer, it attracts the sperms. So this white color line is the zona pellucida, which is surrounded by corona radiata cells or follicle cells. We can see once the sperm touches the zona pellucida, it changes the permeability of the cell membrane. So it attracts the sperm for fertilization. Once one sperm enters, then the zona pellucida alters the permeability of the cell membrane, and it prevents no more sperms entering inside. So only one sperm is allowed usually to enter inside the ovum. So let's talk about the sequence of fertilization. So before spermatozoa can fertilize the oocyte, they must undergo capacitation. So the changes happening in the sperm is called as capacitation. Capacitation occurs within the female genital tract, that is within the cervix and the uterine cavity. Capacitation occurs. Only the capacitated sperm is allowed to enter inside the ovum. So the capacitation, during which time a glycoprotein coat which is present over the sperm and the seminal plasma proteins are removed from the spermatozoal head. So the acrosomal reaction after the capacitated sperm, the next event is the acrosomal reaction. So zona pellucida attracts the sperm binds to the membrane. So here this is a capacitated sperm. So now the acrosomal reaction occurs. So the acrosomal reaction, during which the acrosin and a trypsin like substance which is released to penetrate the zona pellucida. So once the single sperm has fertilized the oocyte, the membrane prevents the polyspermy. So they won't allow any more sperms. So only single sperm is allowed. So the zona pellucida attracts the sperm and the sperm binds to the membrane. Then acrosomal reaction occurs. So acrosome reaction with the release of some enzymes and penetration of the sperm into the zona pellucida, and then fusion of the sperm. We can see the zona penetration of the sperm to zona pellucida is called zonal reaction, and then cortical reaction, release of cortical granules, where the fusion of the sperm with the oocyte membrane happens. So these are the sequences in the fertilization. First is attraction of the sperm. Before attraction, the sperm has to get capacitated. So only capacitated sperm can fertilize the ovum. Second thing is this acrosomal reaction, then penetration through zona pellucida, then fusion of the sperm, then that is acrosomal reaction, then zonal reaction, then cortical reaction releasing cortical granules. So the fertilization is it is like change in the permeability of zona pellucida is the zonal reaction. So it is a multiple sequence where the sperm bind to the corona radiata by only single sperm usually fertilizes the egg. So the acrosome reaction, the sperm bind to the zona proteins and the zona that is in the zona pellucida, which initiates the release of trypsin-like enzymes called as acrosin from the sperm and to burrow the to dig the zona pellucida. Then next is the zonal reaction binding the sperm and the egg plasma membranes initiates because of calcium influx, calcium ion influx into the egg and release the cortical granules from the egg that block the other sperm from fertilizing the egg. So we can see the stages of fertilization. Only one sperm is entered here. We can see the acrosomal cap here. Here the acrosomal cap is removed here. So due to acrosomal reaction, and the blue one is the zona pellucida. So once it enters, this is the cell membrane. Once it enters, only the head enters inside and remaining part of the sperm sheds off. So acrosomal reaction means as the sperm is attracted to the zona pellucida, which is a glycoprotein membrane, it releases enzyme called as acrosin, which allows the sperm to penetrate the zona, thereby coming in contact with the plasma membrane of oocyte. So the cortical reaction, that is also important to know, as the head of the sperm comes in contact with the oocyte surface, that is the plasma membrane of the oocyte, it results in the release of lysozymal enzymes which from the cortical granules which are lining the plasma membrane of the oocyte. So this triggers trigger for this cortical granules to exocytose is the release of calcium ions from cortical smooth endoplasmic reticulum, which is located inside the cytoplasm of the egg. So the smooth endoplasmic reticulum is response for sperm binding to the egg. And zona reaction, in turn these enzymes alter the properties of the zona pellucida to prevent another sperm to penetrate, preventing polyspermy, and inactivate the species specific receptor sites for spermatozoa on the zonal surface. So the sequence of reaction is acrosomal reaction first, then cortical reaction second, and third is zonal reaction. So these things to be noted. So during fertilization, the spermatozoa must penetrate corona radiata. So first it has to penetrate corona radiata, then zona pellucida, that is the second, then third the oocyte cell membrane. So as soon as the spermatocyte has entered the oocyte finishes the second meiotic division. We know that oocyte is arrested in second meiotic division. So it completes its second meiotic division and forms female pronucleus. So the zona pellucida becomes impenetrable for other spermatozoa, preventing polyspermy. The head of the sperm separates from the tail, only the head would be there, and tail and the head swells and forms a male pronucleus. Both the pronuclei have to replicate their DNA, and paternal and maternal chromosomes intermingle and they split longitudinally and go through the mitotic division, giving rise to two-celled stage. So that is how we see the initiation of cleavage now. Cleavage means cell division. So the single-celled zygote will turn to form two-celled stage. So the results of fertilization is, one is restoration of diploid number of chromosomes, that is first thing. Next thing is determination of chromosomal sex. If the two X chromosomes are present in that, then the female baby. If it is X and Y, it is a male baby. And initiation of cleavage. So these are the results of fertilization. So chances of pregnancy in the days near ovulation, this is clinically quite important. So 5th day before ovulation is 5 days before 10% chances, 4 days before ovulation 16% chances, 3 days before ovulation 14%, 2 days before ovulation 27%, 1 day before ovulation then 31% chances to become pregnant, and on the day of ovulation 33 chances. So maximum chances of becoming pregnant is on the day of ovulation, and day after ovulation means there is 0% of chances of becoming pregnant. Next, coming to the cleavage. Cleavage is a series of mitotic division that results in an increase in the cells, so which are called as blastomeres. So the zygote will divide to 2 cell, 3 cell, 4 cell stage, which are called as blastomeres, and which become smaller. The size will become smaller. If you see, the zygote size is here, and all the cells here together also are the same size of zygote. So individually the cytoplasm divides, making the size of the cell smaller. So after the divisions, the blastomere undergo a compaction to become a tight grouped ball of cells with called as with containing inner and outer layers of cells, and this compacted blastomeres form a 16 celled stage which is called as morula. So 8 cell will turn to form 16 celled stage called as morula, and morula will again further forms advanced morula, that is 16 to 64 celled stage. So you can say that there is no fixed number for morula. 16 to 64 celled stage is considered to be morula. Morula means a mulberry fruit, so it appears to be like a mulberry fruit. That is the reason it is called as morula. And in 16 to 64 celled stage, that is in advanced morula stage, we see the compact cell mass. It enters the uterine cavity, and on the day 4, we see the formation of blastocyst. A cavity starts appearing within this morula, where the inner cell mass will settle at one side and the outer cell mass is present in the periphery, and the outer cell mass is called as trophoblast, and inner cell mass cells are called as embryoblast. So we can see the erosion of the wall of the uterus leading to implantation. So blastocyst formation. As the morula enters the uterus on 3rd or 4th day after fertilization, a cavity appears within the morula, and this cavity is called as blastocele or blastocyst. Cyst means a cavity, and the inner cell mass which is formed at the time of compaction will develop into the embryo proper, and is at one pole of the blastocyst, so which is called as embryonic pole, where we see the embryonic cells here. The other end where there are no embryonic cells, it is called as abembryonic pole, and the outer cell mass which surrounds the inner cell mass of blastocyst cavity will form the trophoblast. The uterus at the time of implantation is in the secretory phase, and the blastocyst implants in the endometrium along the anterior or the posterior wall of the endometrium. If fertilization does not occur, then the menstrual phase begins, and the spongy and the compact endometrial layers are shed during menstruation. So the basal layer remains to regenerate. The other layers during the regenerate during the cycle, and during menstruation it gets shed off. And one thing we need to know that this is the advanced morula stage, 16 to 64 celled stage, enters the uterine cavity on day 4, and it becomes blastocyst on day 4. So we see the formation of blastocyst from day 4, and implantation begins on day 6 or 7. So at the day 6, the trophoblast forms two type of cells. The outer cells of the trophoblast will join together to form a jelly-like substance which is called as syncytiotrophoblast, which is the outer. The inner layer of this trophoblast will form a cellular layer which is highly proliferative layer called as cytotrophoblast. Syncytiotrophoblast helps in erosion of endometrial and helps in the attachment in the endometrial of the endometrium of the wall of the uterus, where it helps in the attachment of blastocysts to the endometrial wall at the day 6, which is called as implantation, and implantation progresses to the second week of gestation. Inner cell mass embryoblast cells of the blastocyst, it becomes bilayered during second week of development. So having two layers, two cell types, epiblast and hypoblast. Then amniotic cavity develops on the dorsal side of the epiblast, whereas where the yolk sac cavity forms below the hypoblast cells. Yolk sac cavity is called as the exocoelomic cavity. We can see the epiblast, hypoblast, cytotrophoblast, syncytiotrophoblast. This is the wall of the endometrium, and these are the blood vessels present within the endometrium, arteries and veins. So embryoblast cells. This is the embryoblast cells, which are forming epi and hypoblast cells, and this is the outer trophoblast. This is the cytotrophoblast cellular layer. This one is the syncytiotrophoblast. Next thing we have to know about the hatching of blastocyst. Preparation for implantation is called as hatching. This point to be noted. So hatching of the embryo from the zona pellucida occurs just prior to the implantation. Till the time the zona pellucida will be intact, keeping the cell mass into a compact ball. So occasionally inability to hatch results in infertility, and premature hatch can result in abnormal implantation in the uterine tube. So we can see the zona pellucida is left, and this forms the blastocyst which is released. This is the hatched blastocyst. This is called as hatching. Hatching is the removing of zona, zonal digestion. The oocyte immediately after ovulation, fertilization approximately 12 to 24 hours after ovulation, and stage 3 male and female pronuclei are formed on here we can see all the stages. So here is the fertilizing happens, where we can see the sperms entering. This is the 2-celled stage, 4-celled stage, 8-celled stage, 16-celled stage. So morula containing 12 to 16 blastomeres approximately on 3rd day of after fertilization, and 7 stage here. This stage is the advanced stage, where the advanced morula stage, and reaching the uterine lumen, and approximately 4 days of age. And early age, that is the zona pellucida has disappeared, which is called hatching, and early phase of implantation, that is where we see the blastocyst eroding, the outer layer of the blastocyst, syncytiotrophoblast, erodes the wall of the uterus, and we can see the embedding, which is called as initiation of implantation. So this is the events during first week of development. So implantation, zona pellucida disappears at the end of first day. The 2 polar trophoblast is implanted on the decidual wall of the uterus, eroding the stratum compactum and spongiosum, which later turns to form stratum basale, that is decidua basalis, and this is due to the proteolytic action of trophoblastic cells. Next, we have to understand about this twinning also. Monozygotic twinning means the single zygote, after forming 2-celled stage, each cell will develop into a separate individual. Then it is called as monozygotic twins, which typically occurs during cleavage and blastocyst stages. So where we can see the cleavage and blastocyst stage. This is the blastocyst stage, where we can see 2 embryoblast proper forming monozygotic twins. Let's see some questions. Fertilized ovum reaches the uterus by which day? 3 to 4 days. When fourth day is the time of blastocyst formation. When it is in the morula stage itself, it reaches the uterine cavity. So fertilized ovum forms the morula, which enters the uterine cavity on day 4. So this trigger for the cortical granules to the exocytose in the release of calcium ions from the cortical smooth endoplasmic reticulum, which is responsible for the sperm binding to the egg. And zona reaction, it is in turn these enzymes alter the properties of zona pellicuda and changes the permeability of zona pellicuda, and where to prevent the another sperm penetration, that is polyspermy, and inactivated species inactivate species specific receptor sites for spermatozoa on the zona surface. So this completes fertilization, that is the first week of development, where we saw the cleavage also, blastocyst formation and initiation of implantation. Thank you.